Hello everyone, I'm Charlotte from MDPI and I will be your host for today. Thank you for joining our latest MDPI webinar on vaccines entitled Infection and Presymptomatic Transmission of SARS-CoV-2. This is the first webinar on vaccines based on special issues from our journal of the same name. If you're interested in submitting your paper to the special issue that is linked to this webinar, please check the webinar's webpage. I would like to remind everyone that this webinar is recorded. The recording will be made available as soon as possible and will be posted on the webinar's webpage. The webinar is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us there. Furthermore, certificates of attendance are available upon request to vaccines.webinar@mdpi.com. We can only prepare certificates for those of you registered and watching on Zoom, not on YouTube. Today's webinar is chaired by Dr. Hatem Elshabrawy, who is an assistant professor at Sam Houston State University. His interests lie in the development of antiviral small molecules and antibodies. Thank you, Hatem, for chairing this webinar. The webinar will consist of 4 presentations today given by Dr. Francesco De Sanctis, Professor Dr. Maria Anagnostouli, Dr. Hussin Rothan and Dr. Kathleen Hefferon. Each presentation will be followed by a Q&A moderated by the chair. If you have any questions, please feel free to post them in the Q&A box at the bottom of your screen. And now I hand the floor over to Hatem. Enjoy, everyone.
Good morning, everyone. This is Dr. Elshabrawy from, I work as an assistant professor in the Department of Molecular and Cellular Biology in Sam Houston State University in Texas, United States. And it's my pleasure today to be the chair of this webinar. We have 4 very esteemed speakers, and they will present different topics as it relates to infection and transmission of SARS-CoV-2. I've been working on SARS, for like 12 years now, and my interest, as Charlotte indicated, is in the development of monoclonal antibodies and antivirals. So our first speaker today would be Dr. Francesco De Sanctis, and he works for the Department of Medicine, University and Hospital Trust of Verona in Italy. And he will talk about deciphering the state of immune silence in fatal COVID-19 patients. And our second speaker would be Professor Dr. Maria Anagnostouli, and she works for University of Athens in Greece. And the third speaker would be Dr. Hussin Rothan, and he works for the Department of Biology, College of Arts and Sciences in Georgia State University in Atlanta, USA. And our fourth speaker will be Dr. Kathleen Hefferon, and she works for the Department of Cell and System Biology in University of Toronto in Canada. So it's my pleasure, to welcome everybody, and we'll hand the mic to the first speaker, Dr. Francesco. Welcome, Dr. Francesco.
Thank you so much. Good morning, everyone, or good afternoon. I will just start sharing the screen, so we start. So good morning, everyone, and thank you for this opportunity to share with you our contribution to COVID-19 research. I'm Francesco De Sanctis. I work in the Verona University Hospital, and this is a picture of our group that is directed by Professor Bronte before COVID and during COVID. The project and the data of the project that I will show you are supported by Fondazione Cari Verona and Fondazione Team. Moreover, previous research and preliminary data that was helpful for this project were supported by IARC. And the data and the results that I will show you are the contribution of internal collaboration within the University Hospital of Verona, but also collaboration abroad with the Dr. Ido Amit from the Weizmann Institute of Science, Dr. Pierre Bost and Menot from the Pasteur Institute of France. SARS-CoV-2 is the etiology agent involved in the COVID-19 outbreak that in about a month was able to record more than 30 million cases and more than 1 million of deaths worldwide. The disease onset occurs usually about 5 days from the infection, and in most of the case, the clinical features of the disease is pretty mild, with fever, fatigue, dry cough, and pneumonia. Unfortunately, a small fraction of patients can progress to a more severe stage of the disease that is coupled with dyspnea and need of oxygen flow and eventually entrance in ICU. Moreover, an even smaller fraction of about 5% of patients can face even a more aggressive and late disease stage characterized by ARDS, multi-organ failure, and eventually death. And according to what we learned from this in this year, comorbidities and age represent the higher risk of severe disease. From an immunological point of view, disease progression is coupled with the release of specific cytokines and hyperactivation of myeloid cells, which ends up with the secretion of pro-inflammatory cytokines such as TNF-alpha, IL-6, IL-10, that can also reach very high level, giving the start to our cytokine storm. This is mirrored peripherically by progressive decrease of T cells in the peripheral blood in spite of neutrophilia. And this state of hyperinflammation, especially in monocyte-derived macrophages, is activated by many mechanisms. First of all, a delayed type 1 interferon response by alveolar epithelial cells that are infected by SARS-CoV-2, which result in higher secretion of cytokines and chemokines involved in monocytes recruitment in the lung, such as CCL2, and the secretion of cytokines involved in the activation of those cells, such as interferon gamma, TNF-alpha, which are usually released by activated T cells and NK cells. Moreover, SARS-CoV-2 can directly activate monocytes and macrophages by infection or by linking to specific receptor, which ends up with activation of inflammasome and activation of NF-κB pathway. All these activation signals ends up with high secretion of IL-6, TNF, IL-8, IL-10, 6CL10, IL-1 array, which are very important in pro-inflammatory cytokines. Especially IL-6, which is released by activated myeloid cells, can act both by signaling on T lymphocyte, triggering activation of CD8 cytotoxic T cells and polarization of T helper towards T helper 17 differentiation. Moreover, IL-6 can also act by transsignaling on the endothelial cells by increasing vascular permeability, by increasing the release of VEGF, but also triggering the monocytes recruitment and neutrophil recruitment by CCL2 and IL-8. And according to this, different cytokines are able to trigger specific response, such as antiviral response by type 1 interferons, such as interferon alpha, interferon beta, or pro-inflammatory and dysregulated immune response, such as IL-6, interferon gamma, IL-2, and all these cytokines that we just mentioned before. And according to these drugs that can interrupt this loop of hyperinflammation can eventually support and help the treatment of this hyperinflammation disease, such as baricitinib, which is an oral selective and reversible inhibitor of JAK1 and JAK2 Janus kinases, which has been already used and approved for indications such as rheumatoid arthritis. For this reason, we thought that we could repurpose this drug also for patients facing COVID. And to address this question, we test in 20 patients administration of baricitinib in order to understand how the clinical consequences of the treatment was supporting in the patients the ability to reach to be cured. This off-label treatment was evaluated using laboratory values, including serum concentration of C-reactive protein and oxygenation index, as well as immunological parameters, including serum cytokine level, serology, and quantification of absolute number of different immunocell population from the enrollment to day 7. First of all, we evaluated if the activation of STAT3 was happening during COVID-19 progression. And this was exactly the case in which, indeed, we found in most immunosubset high presence of phospho-STAT3 compared to healthy donor. We were not able to observe activation of STAT1 during COVID-19. More importantly, we found that baricitinib administration was produced a significant inhibition of phospho-STAT3 in T lymphocyte, NK cells, monocytes, and neutrophils. For this reason, we tested if the treatment with baricitinib could improve the conditions of those patients. The study included 88 patients that were treated with either hydroxychloroquine or antiviral therapy as a single agent or in combination according to clinical feature. Within this cohort, 20 patients were treated with baricitinib 4 mg twice daily for 2 days, followed by 4 mg per day for the remaining 7 days. A low dose of 2 mg twice daily for 2 days, followed by 2 mg per day, was maintained for patients older than 75 years of age. Of course, the patients were matched in the two groups according to different parameters, age, sex, comorbidities, and for several clinical feature values. First of all, we evaluated the safety of the treatment, and this was exactly what we observed, because the patients in the baricitinib group did not experience any type of bacterial or mycotic infection or other consequences. But more importantly, we found that patients treated with baricitinib experienced a faster reduction in oxygen flow need and a more rapid increase in the PF ratio compared to the control group. And also that baricitinib treated patients had a higher, better outcome compared to the control group. Indeed, only 5% of patients faced a worse outcome compared to the control group, in which the percentage was way higher. So better amelioration of clinical condition in the baricitinib group was coupled with an increment in absolute number and faster resolution of lymphopenia with increase of lymphocytes, T lymphocytes, B lymphocytes, CD4 T lymphocyte, and CD4 T effector memory lymphocyte, but also an increase in the production of IgG specific for SARS-CoV-2 spike protein in the patient treated with baricitinib. Moreover, we also observed a faster resolution of cytokine production, especially for IL-1 beta, IL-6, and TNF-alpha, that was significant for baricitinib patients, but not for the control group. Taking together this data from and the results of this observational research, our data supports the adoption of a short-term treatment with baricitinib in regularizing the immune landscape in COVID-19 patients. And considering that this treatment can be orally given to the patient outside the hospital, the impact in limiting the negative consequence of SARS-CoV-2 during the pandemic spread may be of utmost relevance for the global healthcare system. However, also a pharma company will provide final results in a randomized clinical trial using a larger patient cohort. So we look forward to these results. However, during our experience of COVID-19 research, we also performed a deep and complete immunolandscape of viral infection by taking in consideration the peripheral blood of healthy donor, mild patient, severe patient, but also the bulk samples from severe patient. And we integrated to this aim data from a single-cell RNA-seq, protein analysis, and functional assays in order to generate a COVID-19 single-cell atlas, but more importantly, to identify biomarkers associated with different severity of the disease and eventually with different outcome. Moreover, we also performed a viral pulmonary landscape. This is how we divided the patients in 3 groups. We included 5 healthy control, 10 mild SARS-CoV-2 patients, and 21 severe COVID-19 patients. At samplings, the patients were divided into 2 groups according to specific features, clinical features, such as requirement of invasive mechanical ventilation and or admission to ICU, and of course, clinical parameters, such as SOFA score, PCO2, PO2, FiO2, and PF ratio. First of all, we confirm what other studies already observed. That means that COVID-19 progression was coupled with a progressive reduction of lymphocytes and erythrocytes in spite of neutrophils. And we also identify cytokines involved in the progression of this disease, such as VEGF, IL-6, and IL-1RA. More importantly, we performed a deep characterization of the immune system and immunocells present in the peripheral blood and in the BAL. We identify myeloid clusters, mostly neutrophils, but also monocytes, M1-like and M2-like macrophages, and lymphocyte clusters such as CD8 T cells, CD4 T cells, gamma delta T cells, B cells, and NK cells. Of course, some of these subsets, such as M1-like and M2-like macrophages, were present mostly in the BAL. Other subsets were present mostly in severe patient or mild patient compared to healthy control. And we will see now this. About neutrophils, we identified 10 different subsets, including immature neutrophils, resting neutrophils, chemokine expressing neutrophils, ICGS neutrophils, Ph3 positive neutrophils, and CD177 neutrophils. However, when we took a look at the proportion of these neutrophils in the peripheral blood, we identify in the resting neutrophils the most common subset present in healthy control that was progressively decreasing according to the progression of the disease, in spite of IgG neutrophils and immature neutrophils that were mostly present in severe patient and mild patient, but not in healthy control. We also confirmed what we observed by flow cytometry. However, in terms of lymphoid proportion and neutrophils proportion, however, we could not find any difference in terms of monocyte proportion in the blood. Moreover, since we performed this analysis of peripheral blood, we did not have enough cells to perform a deep characterization of these monocytes. However, other studies already confirmed that all the myeloid compartment was altered during COVID-19 progression, and with the instauration of an emergency myelopoiesis and a downregulation of HLA-DR in monocytes according to the stage of the disease. However, none of these studies evaluated the myeloid compartment at a functional level. And this is actually what we did. We used the PBNC from healthy donor pulsed and labeled with a probe that can help us to follow the proliferation of those cells, and we mix them together with monocytes isolated from healthy donor or COVID-19 patients under stimulus of CD3, CD28, and we followed the proliferation of those cells after 4 days. We observed that that compared to the healthy control, in which monocytes do not have any immunosuppressive ability on T cells, COVID-19 progression is coupled with alteration of the monocyte compartment and induction of immunosuppressive function that are very powerful in mild patients and less strong in severe patients. However, if we took a look to the severe patient cohort, we observed that the ones that are losing completely the ability to suppress T cells are the patients that are facing the worst clinical outcome. Moreover, we also confirmed that according to the stage of progression of the disease, we observe a progressive reduction of HLA-DR. However, this was not coupled with immunosuppression nor with survival. However, the expression of arginase 1 was the main player in driving monocyte immunosuppression, and the two markers together were able to discriminate the severe patients that were facing the worst clinical outcome. Of course, we also confirmed this data on the LDN supernatant, I mean neutrophils, and we observed basically the same results with the severe patient losing immunosuppressive ability in the neutrophils, the one with worse clinical outcome. Moreover, when we took a look on the lymphoid compartment and lymphocyte compartment, we found markers associated with the worst stage of the disease, especially activation of NK cells and of gamma delta T cell. However, the most interesting results were found in the bulk samples of severe patients, in which we identify a marker related to the survival of patients with the proportion of M1-like macrophages as the better marker of clinical outcome. And moreover, we found in the CD4 T-resident memory and CD8 T-resident memory the subset that were related better with a less severe clinical condition, and in the presence of naive T cells, the patients that were facing the worst clinical condition. So the take-home message of our results is that COVID-19 progression is coupled with a stepwise alteration of both innate and adaptive immunocompartments. Loss of function in myeloid cells mirrors an immunopathological status progressing from immunoparalysis to immunosilence associated with higher susceptibility to that event. ARG1 expression is the key driver of monocyte-induced immunosuppression in COVID-19. Moreover, the two parameters better identify severe patient facing death. Monocytes in terminally ill patients are flawed in different biological response, possibly including the ability to differentiate into M1 macrophages in the lung, as observed in the bulk of this subject. Moreover, we identify in the gamma delta T cell activation a marker of COVID-19 progression, and in the presence of M1 macrophages in the bulk, the parameter of better clinical outcome, and in the CD4 T-resident memory and CD8 T-resident memory, the link with patients that were facing a better clinical condition. So taken together, our data set the ground for the administration of drugs that can repolarize the immune system, especially in the more severe stage of disease progression, such as by the use of desamectasone in severe COVID-19 patients. Thank you so much for the attention, and now I will be very happy to take your questions.
Talk, and we'll be happy to take any questions. You can type it in the chat or feel free to speak up. I have a question here, Dr. Francesco. I would start here. So it has been known for long, for SARS and SARS-CoV-1 and CoV-2. The belief initially was that the cause of the mortality and the disease severity is a cytokine storm that happened in the patients. But there are many reports that came out recently that shows that there is actually an immune suppression, as you are showing, So what are your thoughts about this? Is it like a cytokine storm? It's an immune suppression, or it depends on the stage of the disease? So initially it's a cytokine storm and then goes into immune suppression.
I think this is a beautiful question, because it exactly was the reason why we performed this wide analysis at a single cell level, but also at the functional level, in order to understand what was happening in these patients. I can tell you that the alteration of both myeloid compartment and lymphocytic compartment is pretty common to COVID-19 progression, but also to other diseases. I didn't not mention because I do not have time to show, but our lab already show that in other diseases, such as in cancer progression, we observe a deep alteration of hematopoiesis or myeloid cell function with an increase of immunosuppression in myeloid cells and monocytes. And we also observed that those monocytes are totally different compared to one of the healthy patient or healthy donor. I mean, not only from an immunosuppressive point of view, from a functional point of view, but also from a molecular point of view and a morphological point of view. Indeed, also in these monocytes, we can find the activation of STAT3 pathway and activation of inflammatory pathways that are very similar to what we observed and what we proved as well in COVID-19. So our hypothesis is that different disease can follow different kind of disease can follow the same track in order to alter the immune system function and at the end generate this loop of hyperinflammation that cannot never restore and ends. And so at the end, the immune system can fail to take under control the disease. This is the final result. And in COVID-19 has been proved when they did this clinical trial using dexamethasone that is a kind of drug that, let's say, interrupt this loop and can support the restoration of a correct immune system function. And this is what we also support with our data. And we also observe that according to the different stage of the disease, both myeloid cells and lymphocytes have different signatures. So basically, the same drug probably is not exactly working at both stages of the disease. This is the reason why we repurpose baricitinib for mild patients, because in that stage of disease can support a better, a faster resolution of the disease. But this doesn't mean, of course, that would work in the same way for severe patients that have a different kind of hyperinflammation. And even if they do not show a real cytokine storm, because the levels of cytokines are not similar to the one that are observed in CAR T-cell therapy, for example, but still doesn't mean that the drug would work in the same way for severe patients. So it's not an easy, there is not an easy answer. Probably it depends on the moment of the disease. This is what we think from the data that we got. But yes, I think that a clinical trial, a complete clinical trial in which they compare different stage, a different drug, we can give a final answer.
Thank you, Dr. Francesco.
Thank you so much.
Thank you. A very interesting talk. So any of the attendees has any questions for Dr. Francesco before we move to the second talk? So it looks like we don't have any more questions.
All right, thank you so much.
Thank you, Dr. Francesco. So in that case, we'll move to our second talk with Dr. Maria. She is working at the University of Athens, Greece, and she's going to talk to us today about host HLA genotype and COVID-19 infection. Thank you, Dr. Maria.
Good afternoon from Athens, Greece. I want to thank MDPI for asking to come here and participate. Those difficult times, we are here, we are healthy, while other human beings have been infected or have lost their lives. And my topic will be on host HLA genotype and COVID-19 infection and the impact on immune response, severity of the disease, and clinical outcome and therapeutics, vaccination included. My disclosures. I want to remind some few points on disease course of SARS-CoV-2 infection, although Dr. De Sanctis already have taught a lot of this. Manifestation of SARS-CoV-2 infections may range from asymptomatic infections or mild to severe course of disease with a high risk of fatal outcome. Common symptoms of COVID-19 are shortness of breath, cough, fever, fatigue, anosmia, sore throat, chest tightness, anorexia, myalgias, dyspnea, sputum production, hemoptysis, dyspnea, vomiting, nausea, and abdominal pain. Concerning the CNS involvement, as a neurologist, I would like to say that the CNS manifestations appear to be a combination of non-specific CNS symptoms, non-specific disease complications, and the effects of one had direct viral infection or inflammation of the nervous system and vasculature with a parainfectious or post-infectious mechanism on the other hand. Concerning neurological manifestations, we have two recent papers on this. One of it is since last September, and it comes from 46 centers from France, and they announced as the common manifestations encephalopathy, acute ischemic cerebrovascular syndrome with large vessel thrombosis, Guillain-Barré syndrome, and in 97 patients with available CSF, they found that 2 of them had positive SARS-CoV-2 PCR. Another paper in last October found that the most pronounced symptoms of CNS are headache, impaired consciousness, loss of smell and taste, encephalitis, strokes, myelitis, acute disseminated encephalomyelitis, neurogenic respiratory encephalopathy, silent hypoxemia, myoclonus, neuroleptic malignant syndrome, and Kawasaki syndrome, mainly in patients, in children patients. As Dr. De Sanctis said before, we have here the T cells involvement in SARS-CoV-2 infection. This is a very last paper from Nature Immunology, where with a pronounced title, T cells in COVID-19 united in diversity, they found that at the resolution states, the T cells, the total T cell response, the solid blue line here, if you show my indicator, is stronger and broader in severe disease, with higher antibody production, as you see here, with the gray solid line. And it is shown here that we have the severe disease, and in mild disease, we have a lot of more T8 lymphocytes production. It is very rare, very strange, but it is so. And we have also some questions about the unknown kinetics of the in the acute phase and memory phase. Talking about T lymphocytes, I have to mention that we talk almost about antigen presentation. Antigen presentation means that we have the involvement of the MHC system and the MHC molecules that present the epitopes, the peptides, to TCR and T cells, and thus the T cells are activated and stimulated. This is the very core first step for immune response, that when we talk about T cells, we have to talk about MHC molecules and HLA molecules as well. Another paper here says, as we already heard from De Sanctis, Dr. De Sanctis, that we have a dysfunction in functionality of the SARS-CoV-2 specific T cells in severe course of COVID-19 disease. This is very important. As we heard, we have the polarization in innate and immune T cells as well. And now some few points about T cells in COVID-19, some more points. The prominent changes in the innate immune system, but also marked alteration of the adaptive humoral and cellular immune response, both SARS-CoV-2 specific and global T cell function. As we've heard, we have the production of the interferon gamma, interleukin-2, TNF-A, which suggests a Th1 phenotype that reminds us of Th phenotype polarization in multiple sclerosis immune responses. SARS-CoV-2 specific CD4 T cells levels are higher than those of CD8 T cells, we have already told it. And unlike CD4 T cells, specific CD8 T cells levels inversely correlated with time after onset of symptoms, which may stability of CD4 T cells. Alternatively, antigen-specific CD8 T cells might have been recruited to the lungs as the SARS-CoV-2 replication is higher. The 4 viral structural proteins differ in their ability to induce specific immune among CD4 and CD8 cells, and maybe the relative immunodominance of the spike protein was described. We have maybe the induction of specific immunity, maybe it's attributed to the pre-existing cross-reactive immunity towards common coronaviruses as primary infectious or reactivation. And the lower CTLA-4 expression levels of SARS-CoV-2 specific T cells maybe is compatible with viral control. And patients were able to mount a strong adaptive T cell response with proliferative potential, and the majority of the patients controlled the viral replication. And finally, the B cells, although we have B-cell lymphopenia, we have the generation of antibodies due to the circulation plasmablasts, and maybe the macrophages through the FC receptor maybe increase the inflammation in lung and produced lung injury. Although studies with larger sample sizes are needed to evaluate whether the more pronounced immunity in patients with SARS-CoV-19 may result in higher stability and better protection from reinfection with SARS-CoV-2 in the long term. And as you have heard, we have the hyperinflammatory production of cytokines in SARS-CoV-2. And now I come to my main point. My aim of my presentation is to present the HLA immunogenetics SARS-CoV-2 infection, how they are useful in finding immunodoptin epitopes due by immunoinformatics, how they are used in animal models, how they are used to be studied in ethnic groups, and at the individual level, we will discuss how they play a role, a very important role in the risk for the disease, the severity of the disease, the specific clinical manifestations as the CNS involvement, the morbidity and fatality, and what role they play with age and gender. As we all know from HLA immunogenetics, the HLA molecules and genes are located in the short arm of chromosome 6. This is the MHC, the whole MHC major histocompatibility complex area. And here we see the class 1 molecules. Genes will generate the HLA class 1, which is A, B, and C, the classical ones, and the class 2, which is DR, DQ, and DP. In between, there are some other genes that produce TNF or complement proteins, etc. The nomenclature of HLA are very difficult, and we have here the broad specificities like the DR2, which is associated with multiple sclerosis in most of the Caucasian populations, which are typed with serological typing in late 70s. But further on, they're divided into DR15 and DR16. Further on, with molecular genotyping, DNA-based, we found more alleles, and now we can announce these more alleles with which are emerged every year and are found in nomenclatures worldwide. And the very first 2 digits remind us from the broad specificity, and the other 2 digits the alleles. As I've said before, when we talk about T cells and the immune response, we have to remind of the trimolecular complex, which is consistent of APC, the antigen presenting cell, HLA peptide, TCR, the T cell receptor, and the lymphocyte itself. And this is the trimolecular complex, the immunological synapses. And this is, as I've said, the first step towards immune response and T cell stimulation. When we have an intracellular antigen, then this antigen is proceeded in the cell, and finally the MHC1 molecule presented to the T cells that become CD8 T cells. And when we have an extracellular antigen via endocytosis, this antigen is proceeded and degraded and is presented on the surface of the molecule with MHC class 2 molecules to the CD4 T cells, and then we have another immune response. The professional APCs, the antigen-presenting cells, are dendritic cells, macrophages, B lymphocytes, activated epithelial cells, and non-professional antigen-presenting cells are Schwann cells, like in the peripheral nerve system. Thus, the antivirus-directed cellular immune responses mean that the binding of variant antigenic peptides to certain MHC class 1 or class 2 molecules, alleles, are the prerequisite for the quality of antivirus-directed cellular immune responses. The same happens with MS pathogenesis, multiple sclerosis pathogenesis. My expertise is on multiple sclerosis and immunogenetics on multiple sclerosis and neurodemyelinating diseases. Here we see the antigen-presenting cell that present an epitope that has an homology to a myelin-associated protein, and thus activates the CD8 cytotoxic T cells and CD4 mature T cells that become Th1 or Th2 CD4 cells. Apart from HLA immunogenetics, we have the cytokine immunogenetics. You already heard about cytokines in COVID-19. I will not go further analyzing all these slides. And I come to the very special paper that summarizes a lot of the data I want to show you today. It talks about the mortality in COVID-19 disease patients and correlating the association of major histocompatibility complex with severe acute respiratory syndrome with SARS-CoV-2 variants. First of all, I have to mention that for the Caucasian patients with multiple sclerosis, there is specific HLAs like the HLA DRB1, 1501, and the whole haplotype, this one you see here, that are associated with the disease. And this produces strong interferon gamma levels. Thus, there are some other HLA alleles that produce interleukin 17, some other class 2 alleles, and all these with other haplotypes like 15, I cannot see here because there are our pictures, but some certain haplotypes and certain HLA alleles produce autoimmunity in multiple sclerosis or other autoimmune disease of the central nervous system. And this shows that certain HLA alleles, and like this one at the bottom, the HLA DQ0602 is associated with narcolepsy, which first was believed to be, there is a gene within the MHC region, which is associated with narcolepsy. Now we have found that narcolepsy is associated with MHC system because these very genes associated with narcolepsy are accommodated in the MHC region. Thus, from this very first step of this paper, it's obvious that certain MHC class 2 alleles are associated in part independent of their antigenic peptides with stronger inflammatory responses that are manifested as increased risks to the development of autoimmune diseases. When we had the very first pandemic of SARS-CoV-1 in 2003, a study in Taiwanese patients found that some class 1 HLA with like the HLA-B 4601 haplotype was associated with the severity of the SARS infection. This is very important because it's the class 1 HLA. And in Hong Kong, Chinese patients had a strong association with HLA-B 0703 and HLA-DRB1 0301, class 1 and class 2 as well. So this data gave triggered the authors here to investigate what happens with COVID-19. On the other hand, some individuals with COVID-19 experience symptoms like Guillain-Barré, which is an autoimmune disease and have an autoimmune background, and this is associated with MHC alleles. And the question thus from all this data arises, will single mutations in the wild type of SARS-CoV-2 or the so-called B strain or L strain have an impact on potential MHC presentation? Because as we've said, HLA presents the peptides to the T cells. Furthermore, the authors here found that some only class 2 HLAs like DRB30202, DRB10101, and DRB0401 have an association with SARS-CoV-2 severe CoV-1. This is very important because they found no class 1 to be associated in these populations with SARS-CoV-2. This is very important. And if we have enough time to analyze all this data in this paper, we will focus on this, that DRB101 is a very, very rare allele. And for our neurological point of view, we found it's in very rare neurological demyelinating diseases like neuromyelitis optica or neuromyelitis optica spectrum disorder. Thus, in conclusion, in this paper, it appeared that there were no selective pressure from MHC class 1 alleles for the tested SARS-CoV-2 variants, and most likely there is selective pressure from MHC class 2 alleles regarding the binding of the ORF8 variants, assuming that this mutation may be biologically relevant. And this data underlines the need to examine SARS-CoV-2 variants and MHC associations along with clinical outcomes, a detailed long-time observation with a specific focus on CNS-associated symptoms, chiefly in individuals with an increased risk to develop autoimmune responses, as we have described it before. Functional analysis applying real-world T-cell recognition assays in association with MHC 4 digits typing at the higher resolution level, as we said, in immunogenetics, and is required to determine and go SARS-CoV-2 directed cellular immune responses associated with the clinical expression of severity of disease and relevant management at points, including deaths. Now we come to another point. We have to show how the immunodomical epitopes can generate both antibody and cell-mediated immunity via peptide prediction methodology. This is a paper from last June from Israel, and they tried to find how from the SARS-CoV-2 proteome, how we could conclude to the immunodominant epitopes, and from these to potential immunodominant epitopes from vaccines targets. And they found that 15 potential immunogenic regions of SARS-CoV-2 that explicitly include 25 mapped immunodominant epitopes, which can generate immune responses by both B cells and T cells. And it's very important because now we will go to the other slide and show that it was not enough. They would like to know at the population level if these immunodominant epitopes have a population coverage worldwide. So they found that they analyzed the coverage of HLAs of all the epitopes from the immunogenic regions of SARS-CoV-2 using a format, an immunoinformatic platform for the population coverage analysis tool. And they found that 7 epitopes from 5 immunogenic regions, which cover more than 87 of the world's populations. And thus the combination of HLA's structure and immunodominant epitopes of SARS-CoV-2 gave data for the discovery of vaccines. Here there's another paper from Mexico, from the other side of the Atlantic Ocean, and they tried to find, and again by informatic prediction, antigen presentation for SARS-CoV-2 spike protein that revealed a theoretical correlation for this HLA, class 2 HLA, DRB101, with COVID-19 fatality in Mexican populations from very different regions of Mexico. And it's very important that we see that different HLAs, class 1 or class 2, play an important role in different ethnic groups. And finally, I come to the animal models that help us to find out how these animal models could help us to reproduce the symptoms of SARS-CoV-2 in mice, which mice, this very specific Draga mice, which has HLA-A2, this is a class 1 HLA, and HLA-DR4. And how they did it, this mice was infused with human hematopoietic stem cells from cord blood reconstitute and a fully functional human immune system, as well as engraft human epithelial and endothelial cells sustain the SARS-CoV-2 infection and develop severe COVID-19-like symptoms. This is very important because it produced the human immunopathological responses to SARS-CoV-2. It's very important, and they proposed that the Draga mouse as a novel preclinical tool for studying COVID-19 immunopathology and human immune response to SARS-CoV-2, including events leading to the cytokine storm and concholopathies, as well as for testing of candidate vaccines and therapeutics. And here they concluded that this is a very, very important tool, and it resembles the real SARS-CoV-2 disease from humans. Finally, because I come to an end of my talk, I would like to say something about vitamin D and SARS-CoV-19 disease. As we know from multiple sclerosis, the vitamin D has a straight action on VTRE, which is the vitamin D response element of and acts of the promoter region of HLA-DRB 1501, which is the most strong risk factor for multiple sclerosis in Caucasian populations. And thus, the low levels of vitamin D enhances the expression of HLA-DRB 1501, and thus it helps for the initiation of multiple sclerosis. And additionally, the high levels of sunlight and vitamin D upregulates the production of T regulatory cells that downregulates the pro-inflammatory cytokines production in multiple sclerosis. Likewise, we found this paper from MDPI journal from Nutrients. It is a very important review saying that vitamin D plays an important role in most infections, especially the SARS-CoV-2 infection, not only via the routes I have already shown to you in multiple sclerosis, but also with TEL-RCs in the cells and abriculation, NF-kappa B, etc. It's not, we have not enough time to analyze it. So, saying all this, I want to sum up and say, HLA alleles are a highly polymorphic system and serve as core element of MHC to present epitopeptides and thus activate T cells to either CD4 or CD8 lymphocytes. COVID-19 infection shares features of immunogenetic associations, immune response, and immunopathogenesis with autoimmune diseases like multiple sclerosis. As I've said before, they have similar HLA associations. They have the production of pro-inflammatory cytokines, or maybe even where the cytokines storm, and they have the activation of innate and adaptive immunity as well. And they are reproduced in animal models, and they have the association with low vitamin D3 levels. Many different HLA class 1 and class 2 alleles have been found to be associated with different ethnic groups, as it happens with COVID-19, the same happened with COVID-19, and we have to use this data in order to better therapeutics. And finally, as a main conclusion, I have to say that many clinical features and mainly disease severity of COVID-19 have been associated with specific HLA alleles in different ethnic groups, but in a small scale so far. So we suggest that we need to have more studies that are urgently needed in order to clarify the immunogenetic profile at an ethnic and individual level towards optimization of therapeutic and vaccination as well. Thank you for your attention.
Thank you, Dr. Maria. It's a very, very interesting talk, and it reminds me of papers that have been published on the association of different types of HLA with the severity of Streptococcus pyogenes infection. So different individuals who have different HLA may respond differently to pyogenes, so severe infection versus mild infection. And this is actually an application of what is happening in bacterial infection. So viral infection also have the same thing. So
yes, same thing, same rules.
Yeah, we're looking forward to see, how the studies are gonna teach us, what happens in COVID-19 patients. Thank you very much.
Thank you.
Would like to see if we have any questions for Dr. Maria.
There is a question in the Q&A box, if you can see it.
Yeah, so there's a question here from Martin Gonzalez. The question is, how do you think this HLA distribution can affect the delivery of the new approved vaccine in United Kingdom?
Yes, as we all know, these very days, we have heard about the massive vaccination in the United Kingdom. We propose that because there are a lot of ethnic group citizens in the United Kingdom, they should study better the immunogenetic background of different ethnic groups that are will be vaccinated. This is our notion and our suggestion. It could be nice.
Thank you.
It takes time, but maybe we have a lot to prevent, having in mind the HLA immunogenetic background. It's cost effective. I could say we could genotype at the low level resolution, not at the high resolution level, as a first step, because it's very cheap and we could do it. And find out which patients, which citizens, not patients, which citizens would be better with one or the other vaccination strategy, or with one or the other vaccine.
Thank you. So any other questions? It looks like we don't have any more questions. So thank you, Dr. Maria. It's a very nice talk.
Thank you very much.
Thank you. Now we'll move to our third speaker, Dr. Hussin Rothan from Georgia State University. And his talk is about the widespread of SARS-CoV-2 virus and how it could be attributed to the genetic variation in the virus spikes. Thank you, Dr. Hussin.
Hello, everyone. Today I'm going to talk about the widespread of the SARS-CoV-2 that could be attributed to the genetic variation in the viral spikes. In general, vaccines consist of two components, which is antigen and the adjuvant. The antigen either is part of the pathogen or the whole pathogen, with the adjuvant, which is important to induce the innate immunity to attract the immune cells to the site of the injection, and then these immune cells recognize the antigen and they produce antibodies and also induce the T-cell response. In general, the ideal vaccine should be safe, easy to administer, and also simple and cheap to manufacture, and also provide long-term protection against the pathogen. SARS-CoV-2, the structure of the virus, as you see here, like the genetic material, which is positive strand RNA, covered or wrapped with the nucleocapsid protein. Sometimes they call it nucleoprotein, sometimes they call it capsid protein. And this protein is very important to fold and package the RNA or genetic material inside the envelope. And the viral envelope consists of different proteins, but the most important protein is the spike protein. The spike protein here, this is the 3D structure of the spike protein, which is trimeric protein. There are two main parts in this protein. The receptor binding domain, the upper one, which is like the head here, and the stalk or the stem, which is the membrane fusion part there. In general, now the vaccine development depends on the spikes. Some vaccines, they use only the receptor binding domain, the part bind to the receptors, and then they use the genetic code as RNA vaccine, like messenger RNA vaccine, or as a DNA vaccine, clone in the plasmid, a mammalian plasmid, and they use this plasmid as to inject to the human as a vaccine, or produce the whole protein by the as a recombinant protein, especially by the insect cells. And other techniques they use, like they clone this one in some non-pathogenic viruses, and then they use it as a vaccine. Or sometimes they use the whole vaccine, the whole virus as inactivated vaccine or live attenuated vaccine. Different strategies to develop the vaccine is like some of them already in a clinical trial, and some of them is past the clinical trial, like RNA vaccine. And developing vaccine is like need time, like from 1 year to 2 years to develop successful vaccines, because there are many steps. Start with the GMP processing, like clinical trials, and then FDA approved, and then produce the vaccine in large scale, and later on the administration and the immunity evaluation. Now, as the spike is important, very important for the immunogenic and the produce or the developed vaccine, also the spikes are very important in the virus widespread or then virus infectivity. If we compare like SARS-CoV-1 and SARS-CoV-2, SARS-CoV-1 in 2003, like the total people get infected with COVID-1 is 8,000 people, and the death cases is 774. Whilst COVID-2, right now, there is like 63 million, which is almost 64 million people infected with this virus, and almost like 1,400,000 deaths worldwide. Like it's a huge difference between the SARS-CoV-1 and 2 in terms of infectivity, and we need to know why this difference are come from, where this very big difference in the virus infectivity. Let us talk about little bit about, before go to the details, about the virus life cycle, how the virus enter the cells and replicate inside the cell. Now, a virus either get activated by the enzyme already available on the cell membrane, which is the host cell enzyme, they call it TMPRS. If this enzyme activates the spike of the protein, the virus directly fuse with the membrane, plasma membrane, and release the genetic material directly to the cytosol, and the genetic material start replication here. If the virus less activated and the spike is less activated by the enzyme, then the spikes bind with the receptor. Both of them should be bind with the receptors. Bind with the receptors, and the virus binding with the receptors induce the endocytosis, and the virus go inside the endosome, and inside the endosome there is enzyme similar to the TMPRS, and this enzyme activates the spike inside the endosome and then induce the fusion, membrane fusion, and release the genetic material. Either this shortcut pathway, which is very fast, or this one. This depends. This will affect the widespread of the virus and infectivity. The spike structure is very important to resolve this one and resolve like many studies give details about this spike. Then we see here, like there are two parts, which is S1 and S2, receptor binding part, which is the most important part here, is the receptor binding domain in the green color here. And there is another part, they call it membrane fusion, and contain many domains of the protein. The most important is the fusion peptide here. And there is HR1, and which is very important after the binding happen with the receptor to make like a conformational change inside this part of the stalk, and then induce the fusion to the inside the infected cell. The genetic variation, this is the main topic here. The genetic variation in the receptor binding domain, if we compare like SARS-CoV-1 and SARS-CoV-2, in general, the sequence identity between these two viruses is about 82%. If we go to the viral proteins, we see here like the polymerase and protease, which is the main viral proteins. The sequence identity is too high, it's 96. I'm almost similar. But if we go to the spikes, we see the sequence identity is 76, which is less. That means there is genetic variation in the spikes. And you see here, where is the genetic variation? For this alignment, the amino acid sequence alignment of these two viruses, the COVID-2 and the spikes for the COVID-1, you see here like the difference in the sequence, the black arrows here, exactly the area where the spike binds with the receptor. And this genetic variation produce like 10 to 20-fold higher binding of the SARS-CoV-2 compared with the SARS-CoV-1, meaning that the SARS-CoV-2 has ability to bind with the ACE2 receptor like 10 to 20-fold more than SARS-CoV-1. And this is very important point for the SARS-CoV-2 to disseminate or to transmission from people, like human to human and animal to human. Now, the second main genetic variation in the activation side of the viral spike. Now, the spike is a trimer, but if we take the one monomer, we find here the blue color, the S1, which is the binding domain, the binding with the receptor, and this color, the cyan color, which is S2, which is the stalk or the fusion part. Between these two segments of the spikes, S1 and S2, there are two sites cleaved by the host cell protein enzymes, which is S1, S2 cleavage site. This is in red color here, and S2 cleavage site. This is in pink color here. And after this site, there is this yellow color here, which is the fusion peptide. Normally, in this case, the spike is inactivated. But when these enzymes, when the host cell enzymes cut here and here, the spikes will be activated. If you have a look at this graph, now here the RBD down promoter, but after the cleave, it will be up and it will be like ready to bind with the receptor. It's more able to bind to the receptors comparing with this status. And also this one is important also in terms of the immune cells and then produce antibodies against this one. It's like they call it immune evasion strategy. The spike is high this part, but when it cleaved by the host cell enzyme, this part will be up and bind with the receptors. Now, the main genetic variation here in this part, there is a new amino acid insertion in COVID-2 comparing with other viruses. Like there are 4 amino acids, which is proline, arginine, alanine, arginine. This amino acid insertion here produce a new site of cleavage comparing with the old site or the previous site with the site 2 and S2 here. This in red color, that means the host cell furin, which is enzyme in Golgi, can cleave and the spikes in this site 1, and pre-activated the spikes before the virus leaves, the new virus released from the cell. Meaning that the new virus, when released from the cell, is almost, some of these viruses is almost activated, can bind with the receptors and fuse with the membrane and release the genetic material without using the endosome pathway. This is here a very important point, and like this kind of insertion or amino acid insertion, you can see in bats. You cannot see in bats coronaviruses, which is very similar to the COVID-2, but other coronaviruses, they have similar sequence of amino acids. As I said here, if the virus, now this is the new one, the furin one. Normally, the virus after release, the TMPRS, like COVID-1, after release from the cell, then activated by TMPRS on the cell membrane, then bind with the receptor and fuse with induce the membrane fusion and release the genetic material inside the cytosol. But in case of the COVID-2, the furin in the Golgi pre-activated the virus, and once the virus released from the cell is already pre-activated, is not necessarily to use the TMPRS and directly can infect other cells or infect the same cell. This genetic variation also enhance the virus infectivity. Besides that, besides the polyacidic amino acid sequence insertion, the proline insertion, which is the first amino acid from this 5 amino acid, this proline insertion also is predicted to result in an addition of, they call it O-linked glycan, because when this proline inserted in the sequence here, the other 3 amino acids will be exposed outside, and these 3 amino acids easy to bind with the carbohydrate groups and make O-linked glycans. And this glycans or the carbohydrate group protect the spikes from the immune cells or the to produce like neutralizing antibodies. You see here like the SARS-CoV-2 and BAT RTG13, which is very close sequence to the CoV-2. Both of them, they have similar sequence in terms of the receptor binding domain, but the SARS-CoV-2 have this insertion of the 5 amino acid, but BAT RTG13 doesn't have the similar sequence, meaning that SARS-CoV-2 is more infectious comparing even with the BAT RATG13. It is very close in terms of the amino acid sequence. It's almost 96% similarity. Now, the possible role of inserted proline to create like O-linked glycan and, to the amino acid serine and threonine and serine, and this glycans will like make the like shield and protect the spikes from the immune cells. We call it here immunoevasion. In general, there are 3 steps for the virus activation in terms of the SARS-CoV-2. Either is a cleave before the release from the cell, or activated before release by the furin and Golgi apparatus, or after attachment, then also here can be activated by the TMPRS, and also inside the endosome, there is enzyme, they call it cathepsin L, also can activate the spikes and release the genetic material. That means it's very high possibility for the spikes to be activated and to enhance the virus entry. And once the virus entry very high possibility, that means the virus infectivity will be very high. Also here, the cleave of this, the site between the ribosome binding domain and the other part of the spikes is very important to stand. This part will be very close to the ACE2 receptor and make the binding very possible. Also, there is also activation of the fusion part. In this case, the virus has like very high activity to enter the cell. In conclusion, the widespread of SARS-CoV-2 could be the host cell, and in the insertion of the polybasic sequence enhances the furin reactivation, which is, as I said, in the Golgi apparatus before it's secreted outside the cell of these spikes. And the proline insertion also is high possibility to produce the mucin-like domain, and this structure protect the spikes from the immune cells, as we call it, immunoevasion. And thank you.
Thank you, Dr. Hussin, for the nice presentation. I worked for a long time on viral entry and developing inhibitors for cathepsin L and TMPRSS2. So in terms of the requirement of the furin or the other proteases like TMPRSS2 or cathepsin L, how do you see the importance of each one of them, and what stage of viral entry does each protease work? Because I know that TMPRSS2 is required, for the 3 viruses, SARS-CoV, MERS, and SARS-CoV-2. But in contrast to SARS-CoV-1, SARS-CoV-2 has a furin site,
Yeah.
which is between the S1 and S2 domains. So, it's not really clear, like when you look at different publications, why this furin site is there and how it does it help TMPRSS2, or as you said, it pre-activates the spike, for further processing, or how, you know, what is the role of this furin site?
Yeah, actually, the insertion of this 5 amino acid is very important to activate the virus before the virus release, the new virus release, or the very own release from the cells. And once it's released, it's already activated. That means it can be infect other cells. Even there is the TMPRS, which is the cell membrane enzyme, is not available there. It can be very activated. I mean, the possibility of the virus activation is too high. Either inside the cell before the secretory pathway before release from the cell by the furin, or at the cell membrane by the TMPRS, or inside the endosome after the endocytosis activation and take up the virus inside the cell. Also, there is cathepsin L also can cleave and activate the virus. That means one more step is combining with the COVID-1, which is the furin activation inside the Golgi apparatus.
Thank you.
Thank you.
I think we have several questions here in the chat box. I'm not really sure about the first question. In insert strains that have SG, you know, PAR, AR, is it more higher clinical symptoms than normal strains? Is it reported case? I think I'm not really clear about this question. It's not clear.
The insert strains of the, yeah. I mean, what I understand from this question, I mean, this insertion of this new amino acid to make like the virus activated by the furin, definitely it's affect the virus entry or activate the virus entry. And once the virus entry like be more powerful, more activated, then the viral pathogenesis will be high. And then definitely this will affect the clinical symptoms comparing with the SARS-CoV-1. And you see like the infection of this virus, even when I test this virus and on the cells, the virus replication is very fast because the virus activation is like much higher than the COVID-1. And definitely once the virus the virus entry enhanced by this activation, by this insertion of the amino acid and enhance the binding, then the virus entry will be much more than the normal, and then this one definitely affect the clinical symptoms.
Thank you, Dr. Hussin. So there's another question from Martin Gonzalez. He's saying, do you think that furin deficiency or blocking its expression could be beneficial? So maybe a knockout model could be used to study that?
Yeah, actually furin is very important enzyme to the cell. I mean, if we knock out the furin, it will be toxic to the cell because it's very, very essential enzyme. And the furin has many functions inside Golgi in terms of producing the host cell protein, because the cell needs the furin to manage the cellular protein, important protein for the cell to be alive. If we knock out, will be a little bit toxic to the cells. I mean, it's not a good idea to knock out it. But in terms of the study, if maybe you can try and to knock out or to partially knock down by the siRNA, and we see like how it affects the virus infectivity. It is a good question.
Thank you. I think we have like questions in the chat box, you know, from Simonita Viviani. So is there any explanation why in some individuals who carry the virus, the mechanism of cell entry is not activated? Not sure whether the question is appropriate. He means in asymptomatic carriers.
No, the asymptomatic is not related exactly to the virus entry here. Asymptomatic is like there are like many factors affect the asymptomatic. The first factor, the age, as we see, like the young people, they have the asymptomatic. The old people, the mature adults, they have symptomatic. And right now there is no like clear answer why there is asymptomatic to the young people and there is symptomatic to the mature adult. This adult people, like many, there are also many speculations, like or suggestions, like the immune system may be different, like the concentration or the expression of the ACE2 receptors on the cell membrane is different between the young and old people. Like there are, but right now there is no, for example, solid data or clear study describe why there is asymptomatic and symptomatic. I think it's not very related to the virus mechanism, virus entry mechanism, because it's unique, it's similar between the adult and the other and the young people.
Thank you.
I'm reading another question from Ali Nazar. He's saying is that clear the glycan structure of the glycosylation, the rule of maturation showing their role in masking. Yes, this is like a prediction, because some studies they predict when the protein inserted in this site of the spike, the protein will change the folding of this area. And some amino acid last time before the insertion, it was like inside the domain. After protein insertion, exposed to outside. And this amino acid is more susceptible to make link with the carbohydrate and make the mucin-like domain. This is not proved by a study, but it's just a computational study showing that this amino acid, if they make this shield of the carbohydrate shield, will protect the spikes from the antibodies or the immune cells.
Thank you, Dr. Hussin. So Martin is following up on his question of furin deficiency and if we can knock it out. He's saying if you are interested, they have in the lab 2 conditional knockout mice in CD4 T cells and macrophage and neutrophils for furin studies.
Yes, yeah, definitely. I'm very interested to study this. And yes, because right now I haven't seen like study describe like if the furin is knocked down or partially knocked down will affect the virus replication. And this will be very interesting study, but not totally knock out. It will be maybe a little bit toxic to the cells. And this depends on the cell type, which cell type we use.
Very nice. Thank you, Dr. Hussain. I think we don't have any more questions. So thank you for your time. It's very interesting. Thank you for all the information.
Thank you for inviting me. Thank you very much.
Now we'll move to our last talk with Dr. Kathleen Hefferon from Canada. And her talk is about using plants to fight COVID-19. And thank you, Dr. Kathleen. Welcome.
Oh, hi there. Thank you. As soon as the screen is cleared, I'll put up my presentation. So thank you very much for inviting me as well. And I'm putting a more interesting, an interesting different twist on the topic today. So my name is Kathleen, and I'm actually at the Department of Microbiology at Cornell University. I do have collaborations and work with the Department of Cell and Systems Biology at the University of Toronto, but I understand they're closed right down right now due to a spike in COVID cases in the whole university. There's no research going on. So I am just locked down in the United States at the moment. So vaccines in plants, what's all of this about? Well, the basic concept is to produce vaccine proteins in plants that we routinely eat, such as tomatoes, potatoes, and bananas. Oral consumption of vaccine proteins via edible plant tissue has been demonstrated to elicit a robust mucosal immune response. So why would we make vaccines in plants? Well, the World Health Organization estimates that millions of people, the vast majority of these being infants under the age of 5, die every year from preventable infectious diseases. And in certain places, traditional vaccines can be very expensive and in short supply. And by making our vaccines in plants, we can circumvent the need for syringes, needles, refrigeration and trained medical personnel. All of these are major stumbling blocks for providing vaccines to the poor and remote places. So these photos come from a colleague of mine who works at Doctors Without Borders. And they're just showing that plants can enhance the availability of different types of pharmaceuticals, such as vaccines, particularly to those who live in remote regions of developing countries. So there are a number of plant-made vaccines that have been developed to this date. Most of them have been researched in academic institutions. We initially started with focusing on these major killers of children in developing countries, such as cholera, rotavirus, Norwalk virus, for example. And then we moved forward and different groups have focused on more common or more diseases that we are familiar with in industrial countries as well, such as influenza virus and human papilloma virus. And then more recently, people have focused on these One Health diseases such as Ebola, chikungunya, Rift Valley fever virus, Middle Eastern respiratory syndrome virus, and so forth. And these photos on the bottom here are from a colleague of mine who works at St. George's University of London. Professor Julian Ma makes antibodies to HIV to block transmission of HIV in tobacco plants. He can then send the seeds to African countries. In sub-Saharan Africa still has terrible time with HIV. And these can be used as a microbicide, so they can block infection or transmission, sorry. Now, in case that sounds too space age for you, there actually are companies that have commercialized pharmaceuticals and plants, and this is the first one, Protelix, which is an Israeli company. It was developed several years ago, and their first product was glucocerebrosidase. So this is an enzyme that's for a particular genetic disease called Gaucher disease. And this particular enzyme could be produced in these carrot suspension cells. So you can see these on the left, these are actually 10-foot tall bags of carrot suspension cells. So they're actually making kind of a carrot juice with this glucocerebrosidase produced inside. So patients who have this genetic disease would just have to drink the glass of this carrot juice a day. And this is really kind of pennies a glass. And to have the conventional drug, you know, the old-fashioned way, it would be $100,000 or so US a year to keep a patient going. So this is a very manageable, it's much more manageable using plants as the production platform here. And some of you may have heard as well of a plant-made solution to Ebola virus. So a few years ago, I think it was the earlier 2012 outbreak, there was an Ebola outbreak in Western Africa, and several Americans came back with Ebola infections. And they were given experimental treatments. And one of the treatments was a cocktail of monoclonal antibodies that were produced in plants using a plant virus expression vector. And these patients seemed to recover. So this was like an experimental antibody made in plants that was used at the time. So that you may have heard that in the headlines at some point in the past. Now, besides just generating, transgenic plants that produce vaccine proteins, we also can use plant virus nanoparticles. And on the left, I put a series of very commonly used plant viruses, which are basically peptide or drug display systems where they will, we can put epitopes or whatever on the surface of these virus particles and have them reassemble. And so on the right, I'm showing you here, these plant virus particles can actually enter mammalian cells. It seems that they can get through with a protein called vimentin and get into cells and thus deliver drug cargoes or epitopes, for example. And so this is something that has taken a lot of interest in my field these days. This is just, just explaining a little further. So we can take different types of plant viruses and either fuse to their capsid protein different peptides, or else conjugate drugs to their surface and infect plants with them. And they end up being suitable for use for different types of therapies. So from this, I'm hoping that I've convinced you that plants can provide a unique opportunity to impact global health using this plant biotechnology. So we can either make the vaccine or pharmaceutical protein in the plant and then isolate the protein and purify it, just like a regular conventional drug or vaccine, and provide that to developing countries. Or we can take just a crude extract, for example, lyophilized tomatoes, which are expressing our vaccine of interest or protein of interest, and then resuspend this into a juice or a sauce or even like a ketchup, I suppose, and provide that itself. And this is very inexpensive because we're skipping a lot of purification steps, which are costly. And a third option, which I like very much, is to use this technology itself to and send that to developing countries so that they can develop their own pharmaceutical industries that focus on their own diseases, their own orphan diseases, or diseases which are primarily ignored in industrialized countries. So to conclude this section, just mentioning that plant-made vaccines could slow the spread of major diseases such as HIV, for example, hepatitis B virus, malaria, and even, I didn't really mention in this talk, but even some cancers. We can show that plant virus particles can slow tumor progression in certain cancers. We could also want to mention that plant-made pharmaceuticals are very inexpensive to produce. They don't require necessarily sophisticated equipment, and they could even be produced locally in certain countries to address the specific needs of a geographical region. Now, moving on, a few years ago, back in Toronto, before there was a border closing, I was working on a project that I was collaborating with several people in Egypt. And this was on the MERS coronavirus, which Middle Eastern respiratory syndrome. So a cousin to our COVID-19 of today. And MERS is carried by camels. A lot of camels, a high proportion of camels are endemic, contain MERS virus. They don't, they're asymptomatic, but every once in a while they'll be able to pass this virus to humans, and the mortality rate is quite high. I think it could be as much as 40%. And these maps here are showing you their camels are all over the place in the Middle East and much of the populated world. So these are ancient trading routes, and camels are all over here. And camels are also pets in a lot of these countries. They're used for trade, they're used as livestock, they're also pets. So there's a camel culture in many countries, and you can see that if you add up the population of these regions here where there are, camel cultures, there's several billion people who basically are around camels, and that's not likely to change tomorrow. So it's quite disconcerting to think that this, virus with high fatality could, possibly cause some kind of pandemic in the future. And of course, here we are with our current situation. So this photo here in the top left is just, I was invited to Egypt and went into somebody's backyard, and here are camels in the backyard, just as we have dogs and cats. So it's really very, very popular to have camels around. And I set up a collaboration with Dev Sidhu at the University of Toronto, who was working on producing an antiviral protein to mares. And so what he's done is this purple on the lower left here, he's produced a synthetic antiviral protein. He did this using phage display mechanism. That he's a protein engineer. And this gray protein here is a viral D-ubiquitinase that's made by the MERS virus. And so this, he calls it a ubiquitin variant, could bind very tightly to this D-ubiquitinase and block the virus life cycle. The virus couldn't replicate or do very much. So it basically knocked the virus out. So then I was interested in getting this into cells because he didn't really have a way of doing that. So we set about making plant virus nanoparticles with the ubiquitin variant fused to the external surface of the plant virus, with the further plan to provide this as an inhaler, because these virus nanoparticles can actually enter cells in the lung and either help patients who have contracted MERS, or perhaps even their family members, help block the virus from infecting them. So we set about this. We used a plant virus known as papaya mosaic virus, which we had handy in the lab. And this virus actually can self-assemble by itself, and it actually can self-assemble in E. coli cells. So it's very useful to use. So we produced this, and you can see we have these shorter virus nanoparticles. This is the wild type virus on the left, this electron micrograph. And here on the right, we have our virus self-assembled nanoparticles that work quite nicely, even though they're shorter. And these are in E. coli cells. So while we were working on this and developing this antiviral treatment, of course, we had our current global pandemic with a related virus, COVID-19. So we kind of switched gears a little to start to address this new problem. And there are a lot of other like-minded plant biotech people out there who had the same thoughts. So I'm just going to show you a few of the different types of solutions that have come up in different places for fighting COVID-19. So here's the first one. This is Medicago, which is a company in Canada. It's in Quebec City, actually. And they, after 20 days of the COVID-19 sequence being released, they were able to produce their first virus-like particle. So here on the left is the SARS-CoV-2 coronavirus, and here on the right is the virus-like particle that Medicago made. So they produced this in plants. So this is a plant lipid layer, bilayer, and these are spike proteins, and it's empty. It's an empty virus particle. So it's all made in plants. It's quite fast and it's quite inexpensive. And in fact, the Canadian government, I think, is planning to use this as a vaccine and has ordered up quite a substantial number of doses of this one. Here's another example of how plants can be used. This is a group, again, it's in Ontario, actually, Western University, and this entity, Suncor, are developing spike protein in algae, and they can use this to produce tests for COVID-19. So algae, again, is a very useful expression platform. Another example you may have read about is British American Tobacco. They are using Kentucky Bioprocessing, which is a company to produce massive amounts of this particular vaccine. It's in tobacco plants. And this is going through the clinical trials right now, but it's looking very hopeful. So this will be millions and millions of inexpensive doses. Here's another company. This is Cape Biopharms, which comes from Cape Town, South Africa. and they are producing spike protein for COVID test kits, and I believe they're also making a vaccine. Final one I'm going to show you as an example is from the University of San Diego. And in this research group uses a different type of plant virus. This is cowpea mosaic virus, which has this icosahedral shape, and they're empty virus particles expressing the COVID-19 spike protein on the surface. Then they can be applied to this patch, which then can enter intradermally into patients. So it's very quick and easy. There's no syringe or anything needed. So back to the research that I'm collaborating with in Toronto. I asked my colleague if he could make a very similar antiviral peptide to COVID-19, since he had made one for MERS, and he said, "Oh, I'm on it already. It's basically done." So he's now got ubiquitin variants for SARS, MERS, and COVID-19, and so they're all basically, completing their initial trials right now. And some other work I'm doing with another research group in the Middle East is trying to use plants to prevent or to produce a vaccine. And this is kind of our strategy here. We've learned through the School of Hard Knocks from other plant expression platform groups of what seems to work and what doesn't for them. So we're kind of using their expertise to find out how to best make a nice stable spike protein to be produced in plants. And so here we've made some changes, and it was interesting to hear Dr. Rothan talk about spike protein and mutations. We've added prolines into our cleavage site to stabilize the trimer. Some of our research collaborators have told us that this helped them a lot to produce nice stable trimers of the spike protein in plants. We also removed the transmembrane domain because that seemed to reduce yield of our spike protein in plants. And we replaced it with a K-del sequence, which is a endoplasmic reticulum retention signal. And that seems to allow the spike protein to accumulate in plants more strongly. And we also added a 6-his tag just so we could collect it, purify it more easily. So we're working on this right now, and this is being produced in plants, and we'll see where we get. And we're quite excited about it. And that's basically all I'm going to mention today. And again, it's a collaboration. A lot of this work has been done at the University of Toronto, and I'm here at Cornell right now and kind of stranded with the border lockdown. So thank you very much.
Thank you, Dr. Kathleen. It's very interesting talk because it's in line with my interest in developing therapeutics, vaccines, and all these things. So thank you. It gives us some insight on how different groups are also working on other on developing vaccines, maybe in plants, and other therapeutics also in plants, which is really, really promising. There is a question here for you, Dr. Kathleen, in the Q&A. So Simonetta Viviani, she is asking, do you have any publication on the HIV antibodies you have met at the beginning of your talk?
So this is Dr. Ma's work, and yes, he has some really exciting work. In fact, I'm trying to remember, he has found that he could make his antibodies in plants work better than monoclonal antibodies that are conventionally made. And they're superior, and he thinks that it's the plant side, or perhaps it might be the plant glycosylation that's slightly different that made them bind more tightly and be more efficacious. So these were kind of neutralizing antibodies that could be supplied to block transmission. And they were superior to what's currently available, and they're cheaper too. So it's Dr. Julian Ma, and if she wants to contact me, I can send her publications, or she could look on PubMed. They're just a few years ago, and she'll find them. They're quite exciting to find that. And one thing I didn't mention is often we find that the plant versions will work better than the conventional mammalian, mammalian cell culture versions. And so we're very excited about that as well. Thank you.
Thank you, Dr. Kathleen. So I have a question here. So how soon you think, these plant-based vaccines, that are developed or therapeutics are going to be out, in the market? Or
yeah, so for COVID-19, they're moving right along. The Medicago version, I bet you it will be out. It might be out by midwinter or early winter even. It's really moving along, and I know the Canadian government has invested heavily in that one. So I would imagine very soon. And so, right on the tail of the first vaccines that we've heard of, you'll start hearing about plant-made versions coming out, shortly afterwards. So they still, obviously they still have to go through the same clinical trials and that sort of thing. So we can't just, dish them out right away. And that's the part that's slow, regardless what production system you're using. Other plant-made vaccines, I think are, for other diseases besides COVID-19, are also under development. And I think within the next 3 to 5 years, you're going to hear about a whole assortment. And the slow part, again, is the clinical trials. So there's a handful that will be out soon. And it's just the finishing with the trials that is lengthy.
Thank you, Dr. Kathleen. So I think there's another question here also from Ali Nizari. He's asking any idea around natural plant products against virus, and also do you know if there's any monoclonal antibody, which my understanding is that will be developed for COVID-19 soon. I think there are many, they're in the pipeline.
Yeah, so I think there are plant-made versions of monoclonal antibodies that we'll see out as well. And I'm familiar with research and development between plant groups and other people who have made monoclonals in conventional systems. So I think we will eventually see that, and that's ongoing. There are, I guess there are herbal remedies. I have read about them, and they're different types of compounds that seem to block infection. Yes, I'm going to leave it there because I think some are in publication, and I'm not sure if their authors want me to disclose before the publication is released. So I don't want to ruin their day. But I do know of a few natural compounds that seem to block transmission of COVID-19. Yeah.
Thank you. Any more questions for Dr. Kathleen?
Yes. May I ask a question? Dear Kathleen, this is Dr. Maria Anagnostouli from Athens, Greece. I would like to ask, I will make a statement first. Hippocrates from Greece said that your food should be your medicine and your medicine should be your food. So you say it in a very, very modern way. And I would like to ask, could we use the conventional vaccines in parallel with plant extracts or differently, separately? Could you use them in parallel together or sequentially?
So I think that's certainly possible, and I think there are some examples. Again, I think actually again we mentioned Dr. Julian Ma, and I think he has an example where he might be using a saponin from a plant that seems to enhance immunogenicity, and it's helpful. So there are examples where yes, you can do that. So it is it's really quite an interesting field to explore, and there's lots to do. And so I think there's some initial work that is indicating that's kind of a direction that should be explored further. Yeah.
Yes. what do you think about children? About children with COVID-19, or to avoid for children to be infected from SARS-CoV-2? Should we use blood extracts or vitamins or whatever?
Yeah, that's a really great question. And I'm not sure if I feel knowledgeable enough about children alone. You know, this seems to be a real black box still about children and COVID-19. In my mind, it's not completely clear. I think it's a really interesting idea. You know, I think you were mentioning about vitamin D, for example.
Yes, yes.
And so, you know, there's something going on there. And there's, you know, that certainly isn't going to hurt. You know, my idea is really to yes, use a more modern approach, but to try to make this quick and affordable for healthcare systems and, you know, people, so that we don't end up in these horrible situations where we're seeing all kinds of deaths and illness and economies going downhill. So so I'm afraid I can't really answer about children, but I think it's very interesting and needs to be explored.
Thank you.
Thank you. So does it look like we have any more questions? With that, we come to an end of our webinar today, and I would like to thank every speaker today for their time and their presentation. Very nice, very informative. And I would like to hand the mic actually to Charlotte, you know, if she has any further comments before we end.
I would just like to thank all of you for your time and your interesting presentations. And thank you to those who've attended also. Yeah, big thanks to everyone.
Thank you. Thank you. Have a good rest of your day.
Thank
Thank
you.
you. Bye.
Bye.